Good morning, guests, and welcome to our digital PCR webinar. My name is Declan, and on behalf of Oxford Global, I'd like to thank you all for joining us today. Today, Alexandra Whale, scientific leader at the National Measurement Laboratory, will be presenting on updating the digital MIQE guidelines, Dmiqe 2020. After Alexandra's presentation, we will have a 10-minute Q&A session, so please submit your questions through the chat option on the bottom of the menu display. The full recording of the webinar will be made available to download on our secure website, and our details will be sent to you after the webinar. So without any more delays, I'd like to get started with the presentation. So Alexandra, over to you.
Good morning, everyone. My name's Alexandra Whale, and I'm not a technophobe, but I'm going to give you a talk today on an update of the Dmiqe guidelines, which we've called Dmiqe2020, that were published a few months ago. So the quick webinar overview, I'm just going to talk a little bit about the National Measurement Lab, the NML, who we are, and then digital PCR over the last 7 years, with a review of the new guidelines. And then within this, I'm going to give some examples of the digital PCR applications. So this is the Molecular Biology Lab. We are based in LGC in Teddington, that's southwest London in the United Kingdom. And the NML is the UK's designated institute for chemical and biomeasurements. We are a centre of excellence for high accuracy measurements in inorganic, organic, molecular and cell analysis. And we work across many sectors such as health and biotechnology. This is our nucleic acid measurement lab, and we support the UK by providing confidence in the genetic measurements being made through metrology. So what is metrology? Metrology is the science of measurement, and this is where we link all measurements back to an international standard unit. This allows us to understand the sources of error, enable traceability in our measurements, and therefore improve the measurement accuracy. This is called a traceability chain under ISO 17511. And as we work our way down the chain, we get nearer to the measurement we actually want to make. So in this particular example, I've got a measurement of a patient sample using qPCR in a clinical laboratory. But as we go down this chain, that measurement uncertainty gets larger. And our work at the NML is to try and define the different sources of uncertainty that can be introduced from the SI unit, so that's copies of a nucleic acid in this case, down the chain to incorporate all the uncertainty. And this is how we support the clinical measurements. So we conduct measurement research to improve nucleic acid analysis. So if we think of the Miqe timeline, the original guidelines were published by Buston et al. In 2009. In reality, the Miqe guidelines came late. qPCR had been in widespread use for more than 10 years before they were published. But we were seeing unreproducible results in the publications, but also basic errors in the data analysis. And a famous example here was the 2002 paper supposedly linking the MMR vaccine with autism. The original guidelines had 4 specific aims with an emphasis on reporting. It was to improve the reliability of results and enable review of publications. Within published qPCR data, there was also clearly a requirement to improve the standard of assay development and validation. And in this paper, the Miqe took a form of checklist where there were a number of items to check, and they were designated as being either essential or desirable to be included within a publication to support the authors, but also the reviewers and the editors. Now, in the last decade, there's been a number of updates to this. So in 2010, Buston et al. Produced a paper to look at the practical implementation of the guidelines. There were then a few papers around the 5-year anniversary looking at the impact the Miqe guidelines had had on reproducibility. And then in 2017, there was a paper by Buston and Nolan called Talking the Talk but Not Walking the Walk. And this was where people were citing the Miqe guidelines in their publications, but weren't actually following the guidelines. It's then also crossed into a number of other areas, such as the 2016 paper looking at the identification of clinical biomarkers and also single-cell qPCR. companies have also begun to incorporate these guidelines into their products. And for those who are interested in the history of Miqe, there is a comprehensive Wikipedia entry with a number of references. But in this talk, we're looking specifically at the Dmiqe guidelines. And this is because there are a number of considerations in these experiments that are specifically relevant for digital PCR. Furthermore, if the Miqe guidelines came a little too late, then these were published in 2013 to get in a bit earlier. There were very few publications that were giving detailed information on assay optimization in digital PCR at this stage. There were also a number of assumptions that were creeping in, such as digital PCR could detect single copies in a reaction and that it was calibration free. And so these guidelines were published to try and prevent that lack of reproducibility that had occurred with qPCR. So this leads us to the 2020 publication with the update. So what has happened in the last 7 years to warrant an update? Well, firstly, our knowledge has increased vastly. There are now a large number of papers that have used digital PCR. And digital PCR is used in one or more main areas. So validating pre-analytical processes, so testing for inhibition or looking at extraction efficiencies. Improving existing measurements, such as the identification of rare variants in a sample. And this is possibly the biggest application of digital PCR. Making novel measurements, so the unique partitioning that happens within a digital PCR allows you to do more complex analysis, such as a drop-off assay, but also supporting the existing measurements, such as value assignment of calibrators. And if you remember that traceability chain I showed near the beginning, with qPCR being used as a routine clinical method, digital PCR could be used to assign a calibrator that is used to generate a calibration curve for that qPCR. There's also many more instruments now and instrument formats. Broadly, the partitioning fits into one of two groups, either fixed physical chamber or a water and oil droplet emulsion. dPCR has become cheaper and much more accessible, and is also higher throughput. We now have many more partitions per reaction, and this impacts on the dynamic range. It also impacts on the sensitivity, but also the multiplexing. So the number of targets we can measure within a single reaction has increased. So there are 4 broad applications of digital PCR. And here on the right, I've just got some examples of the output of the different softwares that we've used over the years. And as I said, one of the main applications is the minority targeting, looking at the rare sequence variant in an abundant wild-type background. And up here, I've just got one of our earlier papers, which was looking at a resistant SNV in influenza that conferred resistance to Tamiflu. And what we can see is when there's 1% resistance, we can clearly pick this out, even in an abundance of wild-type that are shown as the green partitions. The other areas are copy number variation, so looking at prenatal diagnostics or gene amplification in cancer, but also accessing the genetic relationship, so looking at linkage or loss of linkage in a sample. And then there's absolute quantification, so the enumeration, how many bacteria or viruses are in this sample. So thinking along the lines of reproducibility, which is an area we're very interested in the National Measurement Lab. In 2017, we published this paper in Analytical Chemistry that was looking at an SNV in a buffered solution. We recruited 21 labs for this and sent them 4 samples containing different proportions of the KRAS G12D SNV in a wild-type background. And all 21 labs were blinded to the nominal values. This was also using a single digital PCR instrument. And this is a result from one of those samples. And what we can see is that 18 of the labs, without any knowledge of the copy number in the sample, were giving us broadly the same result. There's of course 3 outliers that we can see here, but actually these outliers of the reported measurement were caused by incorrect data analysis, so setting of the thresholds to separate the negative and the positive partitions, rather than the actual experiment itself. A second paper on reproducibility that we've produced was back in 2015 with the lead author Alison Devonshire. Again, we had a more limited interlab comparison between 3 labs, but this was using 2 digital platforms. but also we'd made the material more complex. This was actually containing the bacteria of Mycobacterium tuberculosis. So the extraction was also included in the analysis. And what we found between the 3 labs and the 2 different instruments was only a maximum of 2-fold difference. So bear in mind this includes the extraction and only a 20% CV between the different samples. So this led us to a paper 2 years ago, which was looking at the use of digital PCR as a suitable primary reference method. And in this, we used a comprehensive comparison of that G12D KRAS SNV on the different platforms. This led to the first ever submission of a reference method to the JCTLM database for nucleic acid measurements based on sequence. The JCTLM is the Joint Committee of Traceability in Laboratory Medicine, and it's a database that contains higher order reference materials, measurement methods, procedures, and services. How does this fit in with Dmiqe2020? Well, as I mentioned earlier, the issue of reproducibility is needed in interlaboratory comparisons. So this means we've had access to a large number of experts around the world. Together, we wrote the guidelines and formed the Dmiqe group. We are now 32 authors from 26 different institutions that include universities, hospitals, and other national measurement institutes from 14 countries. Furthermore, when we wrote these guidelines, they are supported by the 8 commercially available digital PCR instrument manufacturers. So how does this fit in with the guidelines and the update? Well, one of the first updates we made was an update to the definitions, and we proposed some new terms as well as clarifying what the terms meant. I want to point out one particular example in this screen, which is the reaction and the pre-reaction. The reaction is what is analysed. So ultimately, this is the number of partitions times that partition volume. And this is not the same as the pre-reaction, which is the total volume that's prepared prior to partitioning. The difference between these two is the dead volume. A second update is terminology for the partition fluorescence, which we have referred to as peak resolution, which is the difference in endpoint fluorescence between the negative and the positive partitions, rather than the fluorescent amplitude of the individual partitions that's been used before. Another one to focus on is the higher order multiplexing, and this is the term given when we count more targets based on that peak fluorescence, rather than there are fluorescent detection channels. So the main headings of the checklist remain broadly the same, but what we no longer have is that essential or desirable element. And instead, we're asking for information to be provided and a place where, if it has been provided, where to find it, and then if it hasn't been included, why not? And an example of this would be the primer and probe sequences. If they can't be provided, perhaps it's a commercially available kit, then there's now a space to say why they can't provide it. So this is moving the checklist into more of a set of guidelines and a tool rather than a set of rules. So in the second part of the talk, I just want to run through a few of the key elements that we have focused on in the paper. Here we've got the design and optimization, so we can look at various things like the annealing temperature, number of cycles, maybe a primer probe titration. And I've got an example here of a genomic DNA target with a single assay within the reaction. We have an annealing temperature going from high down to low. And what we can see is that as the annealing temperature decreases, the peak resolution increases. That is the distance from the positives to the negatives. But this is also an example of how robust digital PCR is, where the copy numbers for each of these different temperatures remains largely unaffected. If we move into multiplexing assays, where the assay optimization is more crucial, the peak resolution is key to setting the thresholds to separate the different partitions. So here we look at the SNV example. So this is the KRAS G12D example. And we can see that when we look in the one color plots with the rare sequence being detected in the blue channel, it's not immediately clear how we should classify these partitions. There appears to be a lot of rain. There's possibly 2 negative partitions. And at the moment, the threshold is set as this pink horizontal line. However, if we look at that 2-color plot, we can start to understand a lot more about the reaction. And we can see that in reality, these droplets here that are sitting at this intermediate precision are in fact the double positives that are exhibiting partition-specific competition that's reducing the peak resolution. But by using this two-color plot, we can set those thresholds to classify those partitions correctly. And incidentally, in our interlab study with the 21 labs that I showed you, it was this setting of the threshold with this double positives that was causing an underquantification, where this line was being set a lot higher. For the higher order multiplexing, we can see that we end up with 3 clusters that are happening above the positive threshold line. We can see that the 2 singles and then a double single occur in the 2-color plot. So we can see that there's 8 combinations here of where a partition might sit. This is perhaps easy to see in a 3-color plot where we get the 3 dimensions and we can see clearly the 8 different clusters that occur. A second area is the use of controls. Here we've used a wild-type dilution series that is being used to estimate the false positive rate of the KRAS G12D assay. So here, with only wild-type copies on a 2-fold dilution all the way up to saturation of the instrument, when we look at the G12D, so this is the false positive rate, we can see that the false positive rate is actually dependent on the wild-type concentration in the reaction. Other areas that are important to define are the treatment of the template prior to the digital, such as the denaturation of the RNA or the DNA before partitioning. This can impact on the copy number, but also digestion. We can digest our samples in the reaction either to separate linked targets or to decrease the viscosity of, say, high molecular weight genomic DNA. There's then a nice large area on the data analysis and setting that threshold, but also the treatment of the rate. Here I've got an example of a genomic DNA sample that is on a 2-fold dilution series. And I've got the first plot here showing where we've set the threshold low to include the rain in the positive fraction, or it's set higher to not include it in the positive fraction. And when we don't include the rain, we end up with a 17% systematic bias. That is, we underquantify our sample. So what we are showing here is that it's important to define and look at your data in order to decide how you're going to analyze your data and understand what you're doing with each of your partitions. There's then some information on the partition volume and the partition volume variance. If you use the wrong volume, you could under or over quant. But interestingly, if your partition volume variance is large, you end up with a systematic underquantification. And there's some very nice papers that illustrate that. We also look at the experimental variance, so looking beyond the Poisson error, so not just reporting the error that can be introduced by the Poisson correction to account for more than one molecule in a partition prior to cycling, but also to look at the between experiment variance all the way up to reproducibility, looking at between labs. And I just want to finish with the final talk, part of the talk on SARS-CoV-2. It'd be hard to do a presentation in 2020 without a nod towards SARS-CoV-2. And what we've been working at in the NML is working internationally with INSTAN and CCQM. So INSTAN is an organization that runs external quality assessments, or EQA schemes. And the CCQM is a consultative committee on quality materials. And we work in the nucleic acid working group that's composed of international metrology institutes. And we're currently running a study to use digital PCR to count the number of SARS-CoV-2 viral targets and materials. As a brief snapshot of some of the EQA schemes that we've been involved in, this is an example of how qPCR can give you log differences. So in this example, which was the scheme back in April, 4 samples were measured, so each of this x-axis, with 34 different labs or targets. And in this scheme, predominantly qPCR was used, and there are 8 measurements in here by digital PCR. And what we can see is there's about a 3 log difference in copies per mil reported by each of the labs. In the June-July scheme, we've been working with 2 other international metrology institutes, so PTB, that's the German National Measurement Institute, and NIST in America, to start to estimate the copy number concentrations per mil, looking at the error bars of the standard uncertainty. And we can see that here we're getting a very close agreement that's less than 2 logs difference. And if you remember back to the talk, the slide I gave on the TB work, where our digital PCR was giving us a 2 log difference, here this is clearly improving the 3 log difference here. So I'd just like to finish with a summary. The update is to account for all the developments and the huge amount of learning we've had in the last 7 years. The aims are, as before, really to empower authors and to empower readers to read and design and understand the experiments that have been done, to give greater scientific integrity, to facilitate the replication of experiments, and to provide the critical information that allows us to understand the papers. And I've just got here on the right just the DOI as well as the citation. So I'd like to finish with the acknowledgements. It's a huge number of us in the NML who've been doing a huge amount of work on digital PCR over the last 7 years. Of course, the Dmiqe group, the 32 authors who have published the update, the 8 different commercially available digital PCR platforms we've been working with, as well as the Instand GBD who are running the equa schemes, and NIST and PTB who are working with us to value assign the SARS-CoV-2 materials. So thank you very much for listening, and I will hand you back over to Declan for some questions.
Yeah, brilliant. Thank you very much, Alexandra, for this informative presentation. And so now we move on to the Q&A section of the webinar. So please submit your questions through the chat box on the bottom of your menu display, and we'll get through to them as they come through. So I guess first of all, I have a question for you, Alexandra, anyway. So what do you think are the biggest or recent advancements of digital PCR or DPCR?
So I would say some of the biggest advancements have been just the sheer breadth of the types of assays that are being used. So I mentioned very briefly at the beginning the drop-off assays, but also the higher order multiplexing. We can now start to properly genotype based on the sequence for diverse applications like CRISPR. With the drop-off assays, we can go in and start looking and asking the question, does this patient have a mutation? So something like the EGFR mutations, the wild type has a particular treatment. If there are mutations, you have a different kind of treatment, and it's not necessarily directed on the specific mutation, just the presence or absence of the mutation. And so while normally we would associate that kind of analysis with a sequencing method, we can actually do that pretty quickly with a digital PCR method now.
Okay, yeah, brilliant. Thank you for answering that question. So I guess I have another one as well. What do you think the next 10 years will look like with DPCR?
Well, I'm hoping that the use of digital PCR for value assignment of calibrators that can be then incorporated into workflows that we already have. So we have qPCR being used for a large number of clinical measurements. And if we could use digital PCR to value assign the calibrators that are being used, then that would be brilliant for the reproducibility. Eloise Busby, who's a PhD student in our lab, did a lovely study published a few years ago where digital PCR was used to value assign calibrators in HIV quantification. And when the digital value was used rather than the assumption value of one HIV copy per cell in the calibrator curve, suddenly 3 different labs, rather than having that 2 to 3 log difference in their measurements, were suddenly harmonized to be giving much closer results. And so by using digital PCR to value assign the calibrators rather than doing the actual measurement, we're not asking labs to invest in digital PCR and to change their current workflow, but more to give them an extra tool to support the good measurements that they're already doing, just allowing that traceability down through that chain.
Okay, brilliant. Thank you for that, Alexandra. Okay, just a reminder to everyone, there is the chat box down below where you can enter your questions, so submit them through the main display. I do have a question here as well. So why do I need dPCR if I have qPCR?
Well, this is a question that gets asked a lot, and I suppose it depends on what sort of measurement you're doing. With digital PCR, you have a number of extra requirements over the qPCR, mostly being that partitioning that needs to happen before, and then an ability to read afterwards. Now, qPCR in general is more high throughput, but as I was saying, digital PCR can make the direct measurements itself. And when we're doing something more complex, like looking at evaluating a CRISPR gene efficiency, then perhaps digital PCR would have the edge over qPCR. However, if you're doing routine monitoring, say diagnosing SARS-CoV-2, then real-time PCR with the throughput would give you a quicker ability to measure this, particularly as all of the labs tend to have qPCR capabilities. But it's really the support from digital PCR to make the more niche measurements that is so crucial, particularly with things like cell-free DNA, when we're looking at monitoring and diagnosing patients with the different rare mutations that give us an insight into what their solid tumor might be from taking a blood sample. And that's where digital PCR is really being used, and where I think it definitely has the edge over qPCR, that has a much less sensitive limit of detection, sort of around about the 5%, whilst digital PCR is down at the sort of sub 0.1% level.
Right, so we don't seem to have any more questions currently, so I guess that brings our webinar to a close. Just a reminder, Alexandra is one of Alexandra's collaborators, Jim Huggett, Senior Lecturer and Analytical Microbiology at the University of Surrey, will be speaking at our upcoming Virtual Next Genomics series, taking place next week between 3rd and 6th of November. So if you'd like any further information on the Congress, or would you like a copy of the conference brochure, please contact the marketing department or visit the conference website. And on behalf of Oxford Global, I'd like to thank everyone for joining us today. And I'd like to say a big thank you to Alexandra, who's put a lot of time and effort into making this webinar as interesting and valuable as possible. So I hope everyone has a great day, and we look forward to working at one of our future events.